Welcome to the deep dive. Today, we're really getting into beta blockers, and maybe not in the way you think. We all know them for heart conditions, but their story is, well, it's much bigger. It's about a huge shift in science in understanding how drugs actually work and how our cells talk to each other.
Exactly. It's a fascinating journey looking back at the history and the incredible impact beta blockers had on developing receptor theory, signal transduction, basically how cells get messages.
Yeah, we'll trace it from the early days when things were a bit fuzzy to the kind of detailed molecular picture we have now.
And our goal here, our mission, if you like, is to pull out the key knowledge, those real aha moments.
We want to show how beta blockers didn't just change pharmacology, but they're still shaping medicine. Think of this as your fast track to getting up to speed on some really foundational science.
Okay, let's dive in.
So before beta blockers really arrived on the scene, what did pharmacology even look like? What was the state of play?
Well, before the mid-20th century, it was largely based on observation. You gave a substance, you saw an effect. Simple as that almost.
But you didn't know why?
Not really, not at the molecular level. It was very much a black box. Drug goes in, effect comes out, but what happened inside the cell? That was mostly a mystery.
So lots of empirical data, but not much mechanical understanding underneath it all.
Precisely. I mean, there were some brilliant early ideas, don't get me wrong. You had Ehrlich saying agents only work when they're bound. That idea of needing a physical interaction.
Right. Corpora non agunt nisi fixata.
Exactly. And Langley's receptive substances pointed towards binding sites too. But these were concepts, foundations. The detail just wasn't there. We didn't have the receptor classes we talk about now, GPCRs, ion channels, not formally recognized like today.
So the cell was kind of this sealed unit, pharmacologically speaking, just inputs and outputs.
In a way, yes. It definitely highlighted the need for better theories, but also for better experimental tools to actually peek inside that black box. And the interesting thing is, beta blockers themselves became some of those tools.
Okay, so that's the scene. The black box cell. What ideas were floating around about how drugs might work before beta blockers properly emerged?
Well, there were some key concepts already in place. Affinity, for instance, basically how strongly a drug grabs onto its target. That goes way back to Ehrlich.
Okay, the stickiness factor.
Sort of, yeah. And the difference between agonist drugs that trigger a response and antagonist drugs that block a response that was also pretty well established. The idea of an orthosteric site, the main docking spot on a receptor, was understood too, at least qualitatively.
And someone tried to put math to it.
Yes, A.J. Clark. His work in the 30s was really important. He applied basic chemical principles, suggesting you could analyze drug receptor binding mathematically. This led to his occupational theory, trying to link how many receptors needed to be occupied to get a certain effect size.
So moving towards numbers, towards quantification.
Definitely a step in that direction. But, huge gaps remained. Receptor classification was rudimentary at best. Why did adrenaline constrict some blood vessels but relax others? Nobody really knew.
So classifying receptors wasn't systematic yet.
Not really. And the actual nature of these receptive substances, still unknown. Plus, the massive question, how does binding outside the cell cause changes inside? Signal transduction was a giant unknown. GPCRs, CMMP in this context. Just not on the radar.
So a receptor was more of a concept, a necessary idea, rather than something tangible they could isolate or describe chemically.
Exactly. It was inferred from the drug's action. And while they knew about antagonism, blocking effects, they couldn't really measure how well something blocked. It was qualitative. That's where beta blockers and things like child analysis would really change everything, making receptors feel much more concrete.
Okay, so we've got these foundations, but lots of unanswered questions. Then along comes Raymond Alquist in 1948. What was he trying to figure out?
Alquist was tackling a real head-scratcher. Why did drugs like adrenaline and noradrenaline, sympathomimetic have such varied, sometimes opposite effects in different parts of the body?
Right. Stimulate the heart but constrict a blood vessel. Seems contradictory.
It did. So Alquist did something very methodical. He took 6 of these related amines and tested them across different tissues, carefully noting their potency, basically, how much drug it took to get an effect. He looked at their rank order.
The order from strongest to weakest.
Exactly. And what he found was the key. The rank order wasn't the same everywhere.
Can you give an example?
Sure. For making blood vessels constrict, adrenaline was strongest, then noradrenaline, and isoprenaline was way down the list, quite weak. But for stimulating the heart, totally different picture. Isoprenaline was the most potent, followed by adrenaline, and noradrenaline was relatively weak.
Ah, so the league table changed depending on the tissue. How did he explain that?
His big idea, his hypothesis, was that there wasn't just one receptor type for these amines, but two distinct types. He called them alpha, or aura, and beta, or wuck.
Alpha and beta.
Yep. Generally, he linked alpha receptors to excitation, like vasoconstriction, and beta receptors to inhibition, like smooth muscle relaxation, but also, critically, to heart stimulation. This was the conceptual leap, not one receptor doing different things, but different receptors causing different actions.
That's a fundamental shift. From one target to multiple targets for the same molecule family. How did that go down?
Well, not smoothly at first. His paper was actually rejected initially.
Really?
Yeah, it just goes to show how new ideas, paradigm shifts, often meet resistance. People were comfortable with the existing models. Even years later, it wasn't universally accepted. But the beauty of Ahlquist's theory was that it was testable.
How so?
Well, if there really were two different receptors, you should be able to find a drug that blocks one but not the other. You could find a specific BALT blocker. And that's exactly what Sir James Black set out to do, driven by Ahlquist's theory and a real clinical need.
And finding those specific blockers would prove Ahlquist right.
Precisely. At the time, there weren't any good selective bassanic agonists known. Beta blockers filled that gap and provided powerful evidence for his ion under receptor idea.
Okay, so Ahlquist provides the theory. Enter Sir James Black. This is where we see rational drug design really take off. What does that mean in this context?
It means Black wasn't just randomly screening compounds hoping for a hit. He had a clear goal based on physiology and Ahlquist's hypothesis. He wanted a better treatment for angina, the chest pain from poor blood flow to the heart.
And the usual approach was things like nitroglycerin to increase the oxygen supply, right?
Exactly, increase the supply. But Black thought, well, what if we decrease the demand? Adrenaline and similar compounds make the heart work harder, beat faster, use more oxygen. Black figured if you could block the Brock receptors on the heart, which mediate that stimulation, he could reduce the heart's workload and oxygen need. That was a totally different strategy.
A really clever switch in thinking. Reduce demand, not just boost supply. So how did he find a drug to do that?
It was a process involving a few key compounds. First came dichlorosoproterenol, DCI. Discovered by researchers elsewhere, it was the first compound shown to actually block boreceptors.
So proof of concept.
Yes, huge. It showed Ahlquist's boreceptors could indeed be blocked chemically. But DCI had a big problem.
Yeah.
It wasn't just a blocker. It also weakly stimulated the receptor. It had what we call intrinsic sympathomimetic activity, or ASA.
Meaning it acted a bit like adrenaline itself.
Exactly. So not great for reducing heart workload and not clinically useful. Black actually found it acted as a stimulant in some of his tests.
Okay, so step one, but not the final answer. What next?
Black and his colleague John Stevenson then tried a different tactic. Black called it creating an emasculated hormone. Take an agonist, like isoprenolone, and tweak its structure to remove the on switch, but keep its ability to bind.
Keep the key, but file down the teeth so it doesn't turn the lock.
That's a great analogy. They modified DCI, swapping chlorines for a fused benzene ring, and got prenetholol. This one, launched in '63, was clinically useful. It worked for angina, arrhythmias, and in Black's lab tests, it looked like a pure blocker.
Success.
Almost. Prenetholol worked, but long-term tests in mice showed it could cause tumors in the thymus gland, so it had to be withdrawn. Safety first.
Ah, so close. Back to the drawing board.
Pretty much. But the team at ICI kept refining the structure. They inserted an oxymethylene bridge, a small chemical tweak into the prenetholol structure, and that gave them propranolol, brand name Inderal, in 1964.
Propranolol, that's the big one, right?
That's the game changer. Propranolol was more potent than prenetholol, safer, and developed incredibly quickly. It just transformed heart medicine angina, high blood pressure arrhythmias, preventing heart attacks after the first one. A true blockbuster.
And this work is what led to Sir James Black's Nobel Prize.
Yes. In 1988, it recognized this brilliant fusion of clinical need, physiological theory, and chemical innovation.
Yeah.
A landmark example of rational design, quite different from just stumbling upon a drug.
And the lab methods were key too, you mentioned.
Absolutely. Black's specific assay using guinea pig heart muscle was crucial. It let him see that DCI wasn't just a blocker, but also a partial activator, showing that how a drug behaves can depend on the experimental setup, and you see that clear progression. isoprenolone, the full agonist, DCI, the partial agonist, then pronetholol and propranolol, getting closer to pure antagonists. It perfectly illustrates the emasculated hormone idea.
It's amazing how it all connects back to Ahlquist's original idea. And these beta blockers, they weren't just treatments, they became tools for digging deeper into receptor science itself.
Oh, absolutely. They were far more than just drugs. They were probes, instruments that let scientists dissect how receptors work in ways they just couldn't before.
So how did they help really nail down the existence and specificity of receptors?
Well, like we said, Ahlquist's theory was just a theory until then. But when propranolol came along, and you could give it and selectively block heart stimulation or airway relaxation, the enum effects, while leaving OCC effects like vasoconstriction untouched, that was concrete proof.
You could see the separation in action.
Exactly. It turned receptors from abstract ideas into things you could specifically target and block, tangible.
And they helped quantify things, too. You mentioned child analysis.
Yes, quantifying antagonism. That was huge.
Yeah.
Before, you knew drug A blocked drug B, but how well? Child analysis gave a number to that.
Can you walk us through that briefly? How does it work?
Sure. Developed by Child and Arun Lakshana. Black used it a lot with propranolol. You basically see how much more agonist like adrenaline you need to get the same response when the antagonist propranolol is present. You plot that shift.
Okay, so how much you have to overcome the blocker?
Right. And for a competitive antagonist like propranolol, the quat is a straight line with a slope of 1. This showed they were fighting for the same spot. And crucially, it lets you calculate the antagonist's KB day, its equilibrium dissociation constant.
Which is a measure of?
Its affinity.
Yeah.
How tightly it binds. It gave a hard number to the blocker's potency at the receptor level. This rigorous quantitative approach became the gold standard for classifying receptors and understanding exactly how drugs were interacting, way beyond just it blocks.
So much more precision. And this precision helped uncover even more complexity, like the receptor subtypes, sub-1 sub and sub-2 sub.
Exactly. The first thought was just the MO receptor. But then researchers noticed that some of the newer beta blockers seemed better at blocking heart effects than, say, lung effects or vice versa. This led A.M. Lands in '67 to propose 2 subtypes. Sub-1 sub, mainly in the heart, and sub-2 sub, mainly in smooth muscle like airways and blood vessels.
And drugs were developed that targeted one more than the other.
Precisely. Drugs like Practolol, Metoprolol, Atanol came along. They were more cardioselective, blocking sub-1 sub more effectively. This was great. Clinically, you could slow the heart without causing as much trouble for asthma patients by constricting their airways, a sub-2 sub effect.
So the selective drugs proved the subtypes existed.
Strongly supported it, yes. And later, a sub-3 subtype was found too, mainly in fat cells. The key takeaway is that the frat receptor story established this principle of receptor subtypes, different versions of a receptor for the same natural messenger. And that became a model for finding subtypes in other systems, like Black's later work on histamine H1H2 receptors.
So beta blockers helped confirm receptors, quantify their interactions, and reveal their diversity. Did they also help clarify basic concepts like affinity and efficacy?
Absolutely. They were perfect tools for teasing apart affinity, binding strength, and efficacy, activation ability. Propranolol is the classic example. Binds really well. High affinity, but does nothing to activate the receptor. Zero efficacy. It just occupies the space.
And then there were drugs like DCI, the partial agonist.
Right. DCI showed that activation wasn't all or nothing. It had intrinsic activity, but less than a full agonist like isoprenolone. It could partially turn the receptor on, but it also got in the way of the full agonist.
So it could both weakly stimulate and block.
Yes, which led to the concept of partial agonism. It revealed a whole spectrum of drug activity. Full agonist, partial agonist, neutral antagonist, like propranolol, and even later, inverse agonists that reduce baseline receptor activity. Some clinically used beta blockers, like pindolol, actually have this partial agonist property, ISA. It forced pharmacologists to develop more sophisticated models, like the operational model by Black and Leff, to account for these nuances.
And even more complexity has emerged since then.
Oh yes, the idea of biased agonism or functional selectivity is relatively recent. Some drugs, even some beta blockers like carvedilol, seem to push the receptor to signal through one pathway more than another, say, favoring a pathway involving bororestin over the classic G protein pathway. It challenges the simple one drug, one target, one effect idea. And interestingly, some of those early observations with DCI hinted that signaling wasn't always straightforward.
Wow, so these simple blockers opened up layer after layer of complexity in how drugs and receptors interact, and that naturally leads to what happens next. Inside the cell, signal transduction, beta blockers were key there too.
Absolutely essential, especially for understanding G-protein-coupled receptors, GPCRs, which we now know beta receptors are.
How did they help connect beta receptors to GPCRs?
Well, the way beta blockers worked, by preventing agonists from triggering downstream effects, strongly implied coupling to intracellular machinery. Research eventually confirmed that sub-1 sub and sub-2 sub receptors link up with a specific G protein, GUS. This interaction receptor activating a G protein upon agonist binding is the defining feature of GPCRs. Beta blockers stopped that initial activation step.
So they helped bridge the gap between the receptor binding outside and the G protein activity inside.
Exactly. They linked the pharmacology blocking effects to the biochemistry G protein activation, confirming new receptors were indeed GPCRs.
And this connected directly to understanding the whole KMP second messenger system.
Yes, perfectly. Earl Sutherland had identified KMP, winning a Nobel for it. Beta blockers like propranolol were crucial tools to prove that agonist-induced KMP increases were receptor-driven. Researchers showed add adrenaline, CMP goes up. Add adrenaline plus propranolol, CMP stays low.
Because the receptor was blocked.
Right. But if they bypassed the receptor and added something that acted like CMP directly, they still got the downstream effects, even with propranolol present. This cemented the sequence. Agonist hits receptor, receptor activates G protein activates adenylyl cyclase makes CMP. Beta blockers validated this whole pathway in a vital physiological system.
So they helped map out that crucial first step of the internal signaling cascade. Did they also help figure out what KMP does?
They did. By blocking the KMP production with beta blockers, or stimulating it with agonists, researchers could dissect the consequences. They figured out KMP mainly works by activating protein kinase A, pKa.
Which then does what?
pKa acts like a relay station, adding phosphate groups to other proteins phosphorylating them. In the heart, for example, pKa phosphorylates calcium channels, which lets more calcium in and makes the heart contract more forcefully and quickly. Beta blockers help trace these steps from receptor to pKa to specific protein targets to the final physiological response.
And the subtype selective blockers helped assign effects to sub-1 sub or bus sub-2 sub.
Yes, they help map which pathways were linked to which subtype. And again, the story gets even more nuanced now, with evidence that some beta blockers might influence KMP signaling in specific locations within the cell. This idea of compartmentalized signaling, it's not always just a flood of KMPA everywhere.
It's just amazing. One class of drugs illuminating so much fundamental biology. Beyond the direct scientific insights, what were the broader ripples, the bigger impacts?
Huge ripples. Probably the most significant was cementing receptor-targeted drug discovery as the way forward. Propranolol's success, born from rational design targeting a specific receptor, was a powerful demonstration. It really pushed the pharmaceutical industry towards identifying specific molecular targets first, then designing drugs for them, moving away from just screening stuff randomly. That target-based approach is still dominant.
So it didn't just find a drug, it changed the process of finding drugs.
Absolutely. And propranolol and other beta blockers became essential laboratory tools. Standard reagents in labs everywhere for studying the sympathetic nervous system, adrenergic signaling. Their well-understood properties made them invaluable for research.
And understanding beta receptors must have spurred research into related receptors.
Definitely. Because beta-adrenoceptors were understood in such detail, particularly as GPCRs, they became a model system. The principles learned there, ligand binding, G-protein coupling, second messengers, desensitization, were applied to studying the thousands of other GPCRs. It fueled the whole field. You could draw a line from that early work to the 2012 Nobel Prize for Lefkowitz and Kobilka for their GPCR work.
A direct lineage, and clinically the uses just kept expanding.
Dramatically. From angina, it exploded. Hypertension, arrhythmias, heart failure where they were once contraindicated, anxiety, migraine, essential tremor, glaucoma, even potential roles in cancer are being explored. It just shows how widely receptors influence physiology. And as we touched on, discovering things like biased agonism keeps opening doors for potentially even smarter, more targeted future therapies based on these old drugs.
It's really quite a story, starting from just trying to explain adrenaline's weird effects and ending up revolutionizing pharmacology, treatment, and our basic understanding of how cells work.
It truly is remarkable. Beta blockers really did change everything. validating receptor theory, allowing quantitative drug analysis, revealing subtypes, refining basic pharmacology, eliminating signal transduction, especially for GPCRs and CAMP.
And they firmly established rational, target-based drug design as the way to go, which is still how so much drug discovery works today.
That's exactly right. So maybe a final thought to leave you with. We think of beta blockers as well understood, maybe even simple drugs now. But the ongoing research keeps finding these hidden layers, biased agonism, compartmentalized signaling. It suggests that even these established drugs might still hold secrets, maybe new therapeutic uses we haven't even thought of yet.
It makes you wonder what other established knowledge still has deeper layers to uncover, doesn't it? A constant process of discovery.
Always more to learn.
Thanks for taking this deep dive with us today.